Greetings from National Academy of Medical Sciences, India. Focusing on NAMS Vision 2030, NAMS is pleased to share some of the important activities like NAMS National, International Fellowship and Membership, Member After Day and Beam NAMS, NAMS Task Forces, Aims NAMS Navigate Medico CME, CME activities, publication of annals, newsletter, networking with professionals, public orations, Young Scientist Fellowship, Scientist Exchange Program, scientific visit of Emeritus Professor and many more activities. All application forms for fellowship, membership, associate fellowship, and member after day NB have been made online and objective criteria from the year 2022. NAMS has also started a new online Aims NAMS Navigate CME and Navigate Medico CME program to assist in the development of competencies among PG students and share multimedia learning resource materials as e-library. NAMS has also started 21 task forces to address medical issues of national importance. Under publication activities, NAMS has its own annal as Annal of the National Academy of Medical Sciences. In addition, NAMS also publishes its quarterly NAMS newsletter. For more details of NAMS activities, please visit our NAMS website www.nams-india.in or scan QR codes with the camera app.
Greetings from National Academy of Medical Sciences, India. NAMS has been recognized by the Government of India as a nodal agency for promoting the continuing medical education for medical and allied health professionals. National Academy of Medical Sciences encourages and sponsors nationwide CME programs, symposia and workshops in basic sciences and applied clinical disciplines. In the virtual meeting held on 13 December 2022, more than 200 directors, deans, principals, CEOs, faculty members of the medical colleges and medical institutes decided unanimously that there is a need for the NAMS National Virtual Graduated Medical CME, Navigate Medico CME program for PG students, MD, MS, DNB, MDS. Accordingly, the Navigate Medico CME program was launched on 22 January 2023 by the NAMS President, Professor Dr. S.K. Sareen. Each online session is of 2 hours covering practical aspects as per the needs of the PG students in the identified theme areas. Thank you for your kind support.
Hello and good afternoon, everyone. So welcome to the next session of NAMS Navigate Medico CME program for the postgraduates. So today we are with extremely important topic, and that is the need for pharmacovigilance by our eminent speaker, Professor Y.K. Gupta sir. So to start with the session, I would like to invite Professor Umesh Kapil sir. Sir is Secretary of National Academy of Medical Sciences for the welcome address. Over to you, sir.
Namaskar and greetings from National Academy of Medical Sciences. It's a proud privilege to welcome all of you for our next, or say this, Navigate program. The need for Navigate program was initiated keeping in view of needs of the young postgraduates, need of young faculty members, plus all the medical officers who are in different parts of the country who do not get the opportunity for continued medical education. If we look at our country, we produce almost 100,000 medical graduates, and out of which only 10,000 get into the post-graduation. And a lot of questions are being asked, and there are some of them do not have opportunity to upgrade their knowledge. So the Navigate program is essentially we organize for all the postgraduates, young faculty members, so that they can prepare themselves, they can update their knowledge, plus they can prepare themselves for the NEET examination. We always get the most and the best faculty who can deliver the lecture. These lectures are also put on the NAMS website in a virtual library. All PowerPoint presentations are also put in the NAMS website. Plus, these are broadcasted through YouTube. I understand there are about 169 people have joined on the YouTube. So that makes everyone is comfortable, those who are interested in learning. Today we have with us Dr. Y.K. Gupta. He's a very senior professor with All India Institute of Medical Sciences, and he is the present president of AIMS Jammu. He was dean of AIMS, and I'm sure that you will enjoy his presentation, which is on very important area of pharmacovigilance. Over to you. With these few words, I'll stop over here. Over to you, Dr. Giswant.
Thank you, sir. So next we would listen to the message from Professor Digambar Behra, sir, President, National Academy of Medical Sciences. So, sir.
My dear young colleagues, faculty, and officials of the National Academy of Medical Sciences, a very good afternoon to all of you. With immense pleasure, I invite you all to this program. National Academy of Medical Sciences, that is NAMS India, it was established on the 21st April of 1961 with the objectives of promoting the growth of medical sciences. It was inaugurated at New Delhi on 19th December 1961 by Pandit Jawaharlal Nehru, the first Prime Minister of India. The NAMS fosters and utilizes the academic excellence of health professionals in the country as its resource to promote excellence in medical education and skill development in the field of medicine and allied sciences. The Government of India has recognized the Academy as a nodal agency for promoting continuing medical education for medical and allied health professionals and is advising the Government of India on several matters of national health policy and planning. The best in the field of medicine are part of this umbrella organization, and who is who of medical science guide the NAMS. The National Academy of Medical Sciences encourages and sponsors nationwide CME programs, symposia, and workshops in basic sciences and applied clinical disciplines. The aim of the National Virtual Postgraduate Medical CME is to facilitate the postgraduate students and young faculty members to increase their knowledge in the medical subjects, to navigate them to be more proficient in their profession and pass their NEET and PG examination to become better doctors, eminent biomedical scientists, teachers, and researchers. As a part of this responsibility, NAMS, in collaboration with the AIMS New Delhi and 16 other AIMS-like institutions of the country, has initiated this navigation program in the form of dialectical lectures and discussions. The program is regularly held every first Friday of the month at 3 p.m. The best and useful topics are carefully chosen, and the best teachers from various aims are picked up to prepare and give these lectures. I am sure you all will enjoy these talks and will like them and found them useful in your future lives. Thank you.
Professor Y.K. Gupta sir is a very well-known personality in the field of medicine. Sir is of such a big stature that any introduction about sir would be insufficient. However, I would like to briefly introduce sir. Sir is currently president of AIMS Vijaypur at Jammu. Sir has formerly served as Dean Academics and Head of the Department of Pharmacology at AIMS New Delhi. Sir has also served as President of AIMS Bhopal. Sir worked as former Director CSIR IITR Lucknow, former Founder President of Indian Society of Nanomedicine, former President of Indian Pharmacological Society. Sir developed neuropharmacology laboratory in the department at AIMS New Delhi, and Sir has immense contribution in developing the discipline of clinical pharmacology and strengthening pharmacovigilance programs in India. Sir has also his significant role in strengthening and upgrading National Poisons Information Center, and which is currently functional at AIMS New Delhi. Sir has been bestowed with Excellence Award for Forensic Medicine. Sir is also currently Principal Advisor for India Strategy Development, Global Antibiotic Research and Developmental Partnership, GA Society of Alternative to Animal Experimentation at India. Sir is also National Scientific Coordinator for Pharmacovigilance Program of India. Sir is also chairman of National GLP Technical Committee, DST, Government of India. He is also chairman of RCGM, associated with Government of India. Sir is convener of India Initiate Program, Indian Pharmacovigilance Society. Sir has more than 350 publications in his name in international and national journals. And Sir has guided more than 80 MD, MSc, and PhD students. With this brief introduction, now I would like to request the eminent speaker of today's session, Professor Y.K. Gupta sir, to bestow us with that amazing talk on this very important topic of need of pharmacovigilance. Over to you, sir. Thank you, sir.
Is it okay? Am I? Huh? Thank you very much, Dr. Jaswant, for giving this introduction. Is it okay? Namaskar, and my thanks to Dr. Jaswant for giving such a wonderful introduction. And before I start my talk, I am extremely appreciative and thanks to National Academy of Medical Sciences, particularly, of course, Dr. Behra and Dr. Kapil, Dr. Sood. Dr. Kapil is secretary and Dr. Sood is joint secretary, and for drafting and creating such a wonderful forum for postgraduate and young medical professionals of our country on contemporary topics. I think that this series of this will be of immense value in addition to what they learn and what they are taught in the classrooms, this will be slightly different and increase the employability as well as increase the horizon of our postgraduates. So I am extremely thankful for this. And I am also happy that with the approval of National Academy of Medical Sciences, particularly getting motivation from Dr. Kapil, we have thought of having a series of talks, 8 lectures on pharmacovigilance, schedule of which will be informed to you maybe in next 15 days, and that will be the following topics so that you are fully aware of what is going to be the future series of on pharmacovigilance. One is the pharmacovigilance for industry, because this is one of the most important area in pharmaceutical industry. Each industry is supposed to have one pharmacovigilance team, at least one pharmacovigilance officer, today mandatory. That has a lot of job opportunities also. The pharmacovigilance for public health, and you might have seen that pharmacovigilance became so important during COVID-19 that so many drugs were administered rightly or mostly wrongly, and there were a lot of adverse effects, and particularly you might have seen with antibiotics, excessive use of antibiotics, and that. We will talk on one session on the causality assessment, which is a skill science and which is very crucial for when you make a drug and the side effect association established, because this causality assessment will direct the regulator or the policymaker to withdraw a drug, to allow that drug into the market, to have the label change, and also the precaution, contraindication, indication which is written on that. So causality assessment is something which is very important, and that will be one session. And important thing would be, when we talk of pharmacovigilance, people say, so what? And that's why risk benefit assessment, how you do risk benefit assessment after pharmacovigilance activity, and whether the risk associated with the drug is acceptable or is not acceptable, that how that decision is made. And that is something which is very important, and each industry is supposed to do the risk benefit assessment. The Ministry of Health does risk and benefit assessment. In pharmacovigilance program of India, we do risk benefit assessment if there is a continuous flow of adverse event, and that is how the drug surviving or drug continuation is decided. When one session would be on signal generation, because one has to have the mechanism, and we in Indian Pharmacopoeia Commission, PVPI, we have a signal review committee. I chair that, and in that we decide that how to convert this information into a signal, and then this signal goes to the regulator, and then this is called as whether this drug should be on the focused pharmacovigilance or this should be stopped for the policy decision. In addition, we will have sessions on materiovigilance, hemovigilance, and also pharmacovigilance for devices, which is becoming more and more important. So these are the broad Ruuprekha, or you can say broad sketch of the series of pharmacovigilance program which will follow. With this information, I would give some introduction on the pharmacovigilance so that you are sensitized. If I say that why you develop a drug, new drug, the answer is follows. And that is what all of us know or should know. First thing what we do is this situation analysis. That means if you are developing an anti-cancer drug or anti-hypertensive drug or anti-diabetic drug or simple analgesic drug, you do that what are the benefits and what are the risks or the deficiencies associated with that. Maybe if you are saying cancer, then you can say it is killing the normal cell also. It has a lot of side effects. It has a toxicity. It has a narrow therapeutic window. If you are seeing diabetes, it is controlling the blood sugar HbA1c in a 60% patient, but 30% are not. The drug has to be given maybe 3 times a day. You want to convert into single dose, or you want to reduce the cost. So you do the situation analysis of that. And after this is done, what we do conventionally, we take the molecules and we put this into a computer that is called as a simulation, and then you do a structure activity based or a structure safety assessment. So these are the chemistry based the study you do, and in that more than 10,000 or maybe more molecules are synthesized, and then you conclude with on that basis of the chemistry. And the history says that H2 blockers were developed completely on SAR basis. The calcium channel blockers, the potassium channel opener, the beta blockers. These are the histories where the classical structure activity relation in computer, the penicillins, they were developed after a lot of modification in computer and the structure. And then you come to 10,000, after that you come to 1,000, you funnel them. And what your conclusion you make at the time of funneling of maybe 1,000 or maybe 100 molecules, that with the modification of this structure, based on the background, we conclude that this drug will be effective and we will meet the parameters which we have decided, and this will be safe. And for that we say with reasonable certainty, we say it is safe. Why we say reasonable certainty? Because we have not yet given this drug to rat or mouse or maybe human. Then what you do? The second stage in drug development comes when you do the high throughput screening, enzyme based and maybe receptor analysis, and then again you funnel them from 1,000 to 100, and then you say with a reasonable certainty these 100 molecules or 50 molecules will be effective and safe with a reasonable certainty. And then you go with the regulatory toxicology in different animals, rat, mouse, dog, and whatever the test is recommended by the regulatory authority. I'm not going into the detail, but you may be knowing that. And then when you conclude this preclinical data, this is called as an IND, investigational new drug dossier, and then we go and approach the regulator of the country, and then we say this is the dossier of 10,000 pages, toxicity, safety, and done under GLP, good laboratory practices condition. And now we conclude that this drug will be effective, will meet the decisions which meet the parameters which we have taken in the beginning, maybe efficacy, maybe safe, maybe, and they will be reasonably safe. When you say reasonable safe means because they have been tested in 1 or 2 or 3 or 4 species, they have been tested in a controlled environment, all the animals were given same diet, same weight and similar conditions, and they have not tested in human, and therefore we say with a reasonable certainty. Now, the regulator gives a very cautious direction that knowing that with reasonable certainty this drug is safe to be used in human. That is called as a first dose in human. First dosing in human, and that is the phase 1, and then you go to dose finding studies and the safety in phase 1. Then you go to phase 2, limited number of patients in the disease condition. Then you go to phase 3, the large number, multicentric, thousands of the patients, yet they are controlled. Their inclusion-exclusion criteria, they are almost similar, because ideal situation is not possible to make them exactly similar in their dietary pattern and their environment and their biochemistry and all those things. And then Again, after phase 3 data generation, you go to the regulator called as the NDA, New Drug Application. New Drug Application. And what is New Drug Application? Means you go and the regulator says there, yes, with reasonable certainty based on this data, we permit this drug to be introduced into the market with reasonable certainty. And the drug is now introduced in that particular country where the regulator has given it. That is, the drug is now going to Kanyakumari, to Srinagar, it goes to Bombay, to Guwahati, and east, west, north, anywhere. In a situation where this is not exactly regulated, not homogeneous, maybe old, maybe young, maybe vegetarian, maybe non-vegetarian, maybe genetic difference. Now, if you remember what I said couple of times, the word reasonable certainty, safe, has come right from the beginning. Now, only thing in reasonable certainty, what is certain in reasonable certainty is uncertainty, because it's reasonable certain. And that's why when you say uncertainty, that means when you give drug to million of people, the new side effect will emerge even after, which was not seen in clinical trial, which was not seen or maybe ignored in the preclinical study, or it may be different side effect altogether, it may be in different variation. Maybe you have seen in clinical trial 5% patients suffer from diarrhea, actually it is 20% suffer from severe diarrhea. An allergic reaction you have never seen in clinical trial, but you have seen 2% patients suffer from allergy, and so forth. And therefore, it is important that you must keep vigil on the drugs when they are introduced into the market for new side effects, or as well as the old side effects or the known side effects. And that is what is called as a pharmacovigilance. I was in WHO meeting and there was a lot of debate whether the term pharmacovigilance should be used or it should be called as a medicine safety. There's no much difference in that. But the pharmacovigilance was retained as a term because this gives more powerful sentence, statement, and then you have to keep a vigilance. I will also give you one anecdote, not practicals. My wife is a doctor. When we started pharmacovigilance program in 2010 and attending several meetings, she used to say that why you are going in a vigilance meeting every day? Because this is the job of IPS or police officers and then deputy director administration. That was the level of ignorance of highly qualified in the tertiary care hospital faculty. And this was common. The change of time today, pharmacovigilance has become a buzzword. And the Medical Council of India has made it mandatory for all medical colleges to have pharmacovigilance department, a pharmacovigilance unit. So that is what the changing scenario is. And so with that background, I would say, what are the problem in the pharmacovigilance? And if you just ask in any gathering, have you suffered from any side effect? I think 50% of the population say, yes, I took this. I had a gastric irritation, I had a vomiting, I had a nausea, and somebody will say I had a serious side effect. But then what? Ultimately, this side effect which you have, if you don't collate, if you don't report, this becomes a garbage information. So what is important thing is that you collect this information, collate this information and derive some meaningful information, and then what is important thing is you take a policy decision of that. And that is why this is important, pharmacovigilance is, why this the title of this seminar is, why this pharmacovigilance is important. Because every adverse effect which is happening in one person in Chennai, one person in Delhi, unless this is combined, collated, we will not know the correct picture. I will also tell, particularly postgraduate here, when you read books, and you see that the side effect of this drug is A, B, C, D, E, F, These data, this information is generated from, not from Indian data, unfortunately. This data is generated from US, UK and Japan and those. India is not US, India is not Europe. And therefore, unless we generate our own data, we will depend on that. And I will just give you simple information. Maybe you can do that in your. We did a simple exercise long back, maybe 15 years back. In one group, it was given a tap water or whatever normal water. In other, it was given a packaged water. And what we saw that in India, in volunteers, the people who had packaged water for 7 days had more incidence of diarrhea than those who were taking the tap water normally. What does it mean? That the perception and the evidence are different, and we have to take that the side effect which you see. And today, because of the pharmacovigilance program, we collect more than 1 lakh data every day, every month, and then this is collected. I was just also mentioned, because you must know the background. This is not rocket science. In one meeting in Parliament, there was 7 questions, and one of the major question was an adverse drug reaction. The minister called health secretary, and then it percolated to me. Then I had to attend a meeting with the then health minister in the night. He said, why the drug after so much of testing still cause side effects? And I narrated the same what I told you. And then how can we prevent it? Then he said that, let me know how much money is required. And that was the time when in 2010, I started this pharmacovigilance program from All India Institute of Medical Sciences with 10 centers. And now it has been shifted to Indian Pharmacopoeia Commission for last couple of years. And today we have more than 1,000 centers across the country. We must be proud of this. Is the largest pharmacovigilance program, and we call it the PVPI, Pharmacovigilance Program of India. Another important thing is power of data. There was a time in the beginning when the Uppsala Monitoring Center used to charge $10,000 per year as the fee for putting our data into their program, and they used to give us information. We are good. And today they understood the power of data which India can generate, the population of India, the variation of the population in India, and therefore this data is now we are giving to them free data, and then we get all the information analysis free of cost. And we are also developing our own software which will be used in after this few months. So that is the power of the data is. Why this data is important? Because you take the decision of the drug, it will help the medicine safety, it will help the industry to decide whether this drug to continue or not to continue. It will help industry to change the drug path from one to another. And that is, and to also help the clinician to take the suitable corrective course. This is, I think, background why all postgraduate, all early careers should be aware of. I would just put some of the representative slides because they may not be exhaustive, because I said we will be having a series of that. Now, you just see, this is the fact about the medicine safety. The incidence of drug-related admissions range from 0.2 to 22%. This is the WHO data. 0.2 to 22%, highly variability, because the studies are small, the studies are big, and they are not representative of the globe. You just see, India needs still data to be generated. And there are a couple of people who are working on this, but we have to basically work. What I'm saying, that a couple of people who are using thesis can even work today, that how many in their hospital people are admitted, what is the days number of admission, what is the human resource which is involved, what is the man days lost, what is the health economic impact. These are some burning questions even today. And one can work on these topics. nearly 10 to 20% of data in geriatric hospital admission are related to ADR. There are a couple of publications that after 70 years to 80 years, the average number of medicine per prescription are 5. And the highest number of prescriptions, drug in prescription, 22. If a person is giving 22 medication because he's suffering with diabetes and post-cardiac and arthritis and so on and so forth, there will be a lot of drug interaction, there will be a lot of ADRs which is happening. 10 to 20% of patients are admitted from this. And the data in India, unfortunately, is even not known. So this is something which is very important, and simple topic for any postgraduate working on this is still very important. And I say that if you generate such data, it's less effort, high impact it will call as. And this will be published in a very good journal as well. Now, this is what it says, nearly 7% of medication errors potentially harmful but preventable ADRs. And again, this data, unfortunately, we have to quote this data saying that this is WHO data. Are bhai, WHO data kahan se aaya? This is not from India, unfortunately. What is important thing is we have to create a system where you all work together and find out what is the incidence of medication error. Incidentally, examining the prescription by a pharmacologist is not considered to be a welcome step, but you have to tell the clinician that we are examining the prescription to give you the correct feedback and not to give the deficiency, not to point out deficiencies. And you can see 7% medication error, which are preventable. So if you give the feedback, then this medication error can be prevented. Now, worldwide, more than 50% of all medications are prescribed, dispensed, or sold inappropriately. More than 50%. And recently, because I chair the antibiotic stewardship program of WHO, it is one estimate, I'm not taking that slide here, that the 73% of the antibiotics prescribed were in global data. I don't know whether that had an Indian study incorporated or not. 73% are prescribed in those where the antibiotic was not required. And only 27% were there. Inappropriate means a dose, duration, these are not mentioned properly. And that creates ADR, drug interaction. And 50% of the patient fail to take them correctly. That's called as a non-compliance. Non-compliance. If you do simple study, what is the compliance in diabetes, hypertension, tuberculosis, you know that major problem in tuberculosis is the drug resistance, multi-drug resistance, extensive drug resistance. And the general phenomena where the drug resistance took place was because of the non-compliance, was because of the patient was given medication, was because the patient was not told that it has to be taken for 1 year or 1.5 years. So he stopped taking it after some time. Or if there is any adverse reaction happened, he was not informed of that the urine will become red after taking rifampicin. He will have a nausea after taking few days and it will be correctable. So because of this incidence of ADR, which was not told to him, he stopped medication prematurely and that led to the resistance. And then of course, there are other causes of resistance as well. So this is so important but looks so simple, but has a so important repercussion on that. Increase ADR, adverse drug reaction, the longer the admission in the hospital, longer would be the higher will be the incidence of the adverse drug reaction. And that's why people say, reduce the hospital admission. And decreased compliance and drug resistance. This is the drug resistance, particularly when we talk of antimicrobial resistance, which is also linked to ADR, part of that, is now achieved. Now it is called as a AMR, is a silent pandemic, hidden pandemic. And now all countries, including the G20, with one of the major agenda was this AMR. And in India, for your information, 7 ministries have joined together. Ministry of Health, DHR, DBT, DST, Environment, under Ministry of the Principal Scientific Advisor, where the Ministry of Health is the primary nodal agency to fight for antimicrobial resistance, and also the ADR into that. Now, what is the scope of pharmacovigilance? I'm not going into the detail what is the pharmacovigilance, which I've said. It is new drug and spontaneous reporting. There are two things. One is, if you introduce a new drug, that means, what is a new drug? A new drug is introduced into the market. And then, if you are aware of, otherwise I will just tell you, when a new drug is introduced into the market, after clinical trial, a term is used called as a PSUR, Periodic Safety Update Report, which is mandatory by the industry who has introduced this drug to submit the PSUR data to the regulator. What is that? You have to collect the ADR from India, from entire globe, whatever the data is coming to them, and then you compile them, line listing, and then you submit it after collation every 6 months for first 2 years, and then every year for next 2 years. That means for 4 years, 1, 2, 3, 4, 5, 6 reports have to go. Every time regulator examines this with the help of experts and say, yes, this drug should be continued into the market. Yes, this PSUR should be extended because the drug is still uncertain, or this drug should be corrective. Though that is the PSUR. People say that why this 4 years? Often you are not clear. This 4 years is not a sacrosanct, but if you just see, when a child is born and when he is 1 year or maybe 2 years, he is more prone to do destruction in somebody's drawing room. And so you are more careful when you go to some friends or maybe uncle's place, that he doesn't break the glass, he doesn't break the toys, the showpiece. And therefore, because you do not know that what will be the uncertainty of action of the new drug, and therefore you are more vigilant. More experience with 1 year, 2 year, 3 year, 4 years, you know that now this is established. Now we know that reasonable certainty is now has more. So then you stop this PSUR. And therefore, if the child is not, if the drug is still behaving erratically, the PSUR period can be extended to 5 years, 6 years, 7 years, 8 years. There are incidents where the PSUR was increased to 8 years. Then you will say, why it was not withdrawn? Because the risk-benefit assessment is always done. And if it's still useful, then it is continued. So that is called as a new drug. So the new drug definition is continuous new drug for 4 years. That is the reason why any drug which is new drug is called as a new drug for 4 years. After 4 years, the new drug is regulatory language. It is not called as a new drug. Spontaneous reporting means I am taking any medicine for maybe 340 medicines in national list of essential medicine, or maybe 800 medicines, or maybe 10,000 combination of medicine every day. Spontaneous reporting means if there is anything happens to anybody, he reports spontaneously. So there are 3 types of reporting: spontaneous reporting, and then the elicited reporting, and focus reporting. Spontaneous reporting is the pharmacovigilance program. Anywhere adverse second, you report it. Or when you say focus reporting, that I know that this drug is causing maybe systemic or maybe causing SLE, or in some it is causing Stevens-Johnson syndrome, or maybe fixed drug eruption. And now, when I know that this drug is cause signal generation, fixed dose eruption, then I ask all the centers to see whether there is a fixed dose eruption in such cases, whether there is a constipation. So we do focused eliciting. So this is type of reporting. Medication error and traditional use of medicine is another challenge. In India particularly, when there is a multimodal system used where you are giving traditional medicine also with the allopathic medicine. You are giving in cancer, in epilepsy, in diabetes. People used to take number of other medicines also together. There could be adverse reaction, but we do not. It's very difficult to find out whether it is because of the which component, and that's why it is also important to use that. Herbal, traditional, and complementary medicine, and substandard medicines and counterfeit medicines. This is also within the scope of pharmacovigilance. Don't consider that this is out of that. And in India, the substandard medicine is a big challenge. And what is substandard means? NSQ. This is called as a NSQ, regulatory language. It is called as a not of standard quality. Counterfeit is different. Falsified is different. NSQ is not of standard quality, means it is given as a 250 milligram, it does not have 250 milligram, it is maybe 500 milligrams dose, it is written as 500, it has a 300, or there is some impurities, or there is not dissolve, it doesn't dissolve. So all those are the substandard, but it is a medicine, but it's substandard. Counterfeit medicine, it doesn't have medicine, it has a haldi, it has a chuna, or maybe it has a mitti. So that is a counterfeit medicine, it is a falsified medicine that is usually done for economic purpose. Blood products, now this is something which is very important. Now we do not have the data till few years back that how many incidents of severe allergy has taken place. What is the incidence of other events which have happened for transfusion of the blood or blood products, or even simple saline, and we say that yes, if you have that, how many patient has chills and rigors after the saline administration, we can find out what could be. The new area which particularly all the young generation must look for, the medical devices, because large number of medical devices are being made in India. Now we are becoming independent in medical devices, several programs. Now these medical devices can produce side effects. Even you have a hip replacement or knee replacement, or maybe intraocular lens, or you may have even the cardiac stent, then these medical devices can also produce side effect, adverse effect. And you must know that Johnson & Johnson, which was a major manufacturer of hip joint, the very high penalty was given to them, and all those who were giving Johnson & Johnson hip replacement, they were supposed, they had a re-surgery, and they had, because of the erosion of the metal suspected in those, and then it was several committees we did, and then very high compensation. So medical device, the pharmacovigilance or the materiovigilance, which is called as, or pharmacovigilance device, is very important. Vaccines, we also have the vaccine AEFI, that is adverse event after immunization, called as AEFI. And you might have seen even today, when the COVID vaccine was introduced, people are talking today that those who have received the COVID vaccine, the cardiac incidents are more, this incidence is more. So we have to work whether this is a myth or this is a reality, what is the evidence. And that's why the ICMR has conducted a study, though it was not found to be related. But what I am saying, that if there is an incidence of vaccine related adverse event, this is a mandatory now requirement by regulation, it has to be reported. But for incidents, for other drugs, this is not still the regulatory requirement, but for vaccine, this has to be regulated. So that is again a pharmacovigilance of vaccine is important. Now this is what I call as, this is not given in the books, but I term this as a translational pharmacovigilance. What is translational pharmacovigilance? If there is an adverse event in me, if I have taken say atorvastatin, and I find that my legs are paining, this is the data. Is this data useful? This is the datum is single data. No, it is not useful, because I have to see whether this happens in others also. And therefore, I have found that this happens to me, and I found that this doesn't happen in Dr. Kapil, this happened in Dr. Jitendra, and this happens in Srinagar. Then the signal is generated, and then data is created. Data is plural. Is this data is useful? I say it is partly useful, otherwise it is useless. I say that yes, it has cost in 10%. Now, so datum to data, and then data, unless this data is converted into information. And that's why you talk about AI. Large data is useless. It is junk. It is confusion, unless this is converted into information. That's what I say. This is a noise. Data is a noise, unless this data is converted into music, which gives some information. So information is, you generate information. What information? That atorvastatin, rosuvastatin, and these statins, they cause muscular pain, more in dose related. 10% dose it is less, 10 milligram it is less, 20 milligram is more, 40 milligram it is much more intolerable. And this happens more in female, this happens less in male. So these studies are generated, and then you come to this information. Is this information good? I say no. This information is still not complete, good, because this information has to be converted into knowledge. So datum to data, data to information, information to knowledge. And what is knowledge? That knowledge is that this drug goes into the muscle and interact with the myosin and causes the side effect, which is preventable by reduction in the dose, which is preventable by giving atorvastatin has more, rosuvastatin is more, or whatever the comparatives. We have alternatives available, and therefore you should give this in this appropriate manner. Is this knowledge good? If I ask, had you been present here physically, most of the people will say yes. Now, this is also not 100% useful unless this knowledge is converted into wisdom. And what is wisdom? Wisdom means when you are practicing this, giving these drugs, then you know that if what is likely to happen to this patient, when the physician knows that when he is prescribing that if he gets an adverse effect, how to prevent it, how to mitigate it, risk assessment, risk minimization, risk assessment, and then risk mitigation. And if he knows, suppose you are giving an anti-arthritis drug, or maybe high dose of steroids, then peptic ulcer, then you give, what is mitigation? Mitigation is you give prostaglandin analogs together, you give proton pump inhibitor together, and you avoid giving this. So this is the wisdom which you apply in clinical practice, leading to less adverse effect, leading to better therapeutics, and that is called as a translational pharmacovigilance. From research, data generation to actual in practice, that is what the aim of today's thing. So that when you teach, when you you practice this pharmacovigilance practices. And I will also tell you, those who are in pharmacology may be remembering better that the major side effect of carbamazepine, which is a very effective, cost-effective, very cheap and largely used anti-epileptic drug. Major side effect in Indonesia, Japan, and those areas were 20% patients of carbamazepine has Stevens-Johnson syndrome. In India also they have. The incidence has yet not been known, but very frequently. And of them, 5 to 6% were fatal. They die. Now just think about it. The death happening because of Stevens-Johnson syndrome, because of carbamazepine. So what was done? It was done the genetic analysis, and it was found in the HLA specific gene is involved in this. What was done? That then this gene was isolated, and now if there are 5 people or 20 people of epilepsy, and I will decide whether the carbamazepine should be given or not, before I will give them this genetic analysis. And this chip was created to decide before this, and therefore 90% of carbamazepine induced Stevens-Johnson syndrome could be prevented by avoiding carbamazepine in those patients who are found to have this gene. We did that same study in All India Institute of Medical Sciences, but the carbamazepine kit is yet to be made affordable before it do. So that is I'm saying this is translational pharmacovigilance. This is predictive pharmacovigilance. This is called as a pharmacogenomics and pharmacovigilance. We will talk on pharmacogenomics and pharmacovigilance maybe later, which is useful for drugs of narrow therapeutic window. You identify the gene involved that and identify the population which has that gene and then avoid this drug, or at least you will have the possible impact what they're likely to have. Now, this is what you say genomics, a label advice on and so that's why in genetic variation and the 30% of mild standard. So you know that even warfarin, you know that this population will have large side effects and this population has less side effects. In their drugs like warfarin, which is very important, you do this genetic and now you must be knowing that increasing number of gene analysis facility are being created in major centers. Recently, we inaugurated genomic sequencing lab at All India Institute of Medical Sciences, Jammu. There is a couple of genomic analysis. So now when this genomic facility will be available to you, you will be able to be doing better. So this is one of the very important project which you can think of. ICMR is liberal in funding genomics in ADR. Now, this is also genomics, ABACA, VIR, and then HLA, B. So we are now finding more and more genes involved in causing specific side effects. And that is where it is important. And including that, when you are talking thalidomide, which gave birth to this, still that can be given if you know that which drug. Carbamazepine, I just say HLA-B 1502. Which is in Asian, this is more, and this is in what we recommended in our that this study should be done. I will just say what are the pharmacovigilance challenges in India. It's not easy because reporting, spontaneous reporting is very difficult. People have to be educated. People have to be motivated. Every physician will say, why I should report? A clinician who is sitting in the clinic in the private practice, he says that if I report adverse drug reaction, people will blame, say that this person has causes more side effects, and this will may adversely impact. This is a global phenomenon. But then we decided that how to motivate them and give them awards, we give them recognition. Spontaneous adverse reaction, long-term safety data can be captured through SAE. Serious adverse event. We will talk about the detail about the serious adverse event. Adverse event is one. Serious adverse event means when there is 6 criteria of serious adverse event: death, if there is a occurrence of the permanent disability, there is a increased hospitalization, maybe routine hospitalization in that case maybe 5 days, but this has gone to 15 days. The teratogenic effect. So these are these SAEs. SUSAR is not SUSAR. SUSAR is suspected, unexpected, serious adverse reaction. Remember, this adverse reaction. Suspected, because there is not other thing. So you suspect that this could be. But this was not expected. So it is unexpected because in clinical trial, in the data, you never expected this will call serious adverse reaction. So this is mandatory in various countries that every SUSAR must be informed, because then this SUSAR will become signal, and then this will be put into the what you call as a product information brochure. Safety data obtained through the clinical trials are from controlled situation. And therefore, it is important that to generate everything. Now, I will just give you one idea. This is the anyway, process of pharmacovigilance. And I call them as a 5C. Somebody may say coin 6C, but 5C, or maybe you may call it easy to remember. What you first do is you collect, collate the adverse drug reaction. And AE, when you say adverse event, suppose there is a I am in a clinical trial and then anything happens, it is not necessarily related, so it is adverse event. But when it is causality established, then it is called as a adverse drug reaction. So that is what is causality assessment or analysis. The causality assessment means if this is a drug, if this is a reaction, you have to establish the causality assessment, and that can be done by different methods. There is one which is called as WHO method, there is one method called as a Naranjo classification, and so on and so forth. And I will talk about that. Collate and code the database. Why I'm saying code the database? Because if you collect somewhere that if somebody has taken aspirin or maybe some drug and the adverse event which has happened is मीठा मीठा दर्द हो रहा है, बहुत ऐसा दर्द हो रहा है कि सहा नहीं जा रहा. Now there is no terminology for that. So you have to code that. And then because the data from different language, different countries, the patient may have expression in a different way. Therefore, you have to collect it and then code this so that it is easy to collate. Compute risk benefit. Risk benefit assessment is always, for example, those who are aware of rofecoxib, which is was one of the highest selling COX-2 inhibitor as an anti-inflammatory drug about 15 years back. Highest selling, the one single drug which was highest selling tablet was rofecoxib. Then what was found? That it caused cardiac events. Not in India, in US or Japan or in some European countries, and therefore it was banned. In India, we did not ban it. We had several committees, and I chaired one of few committees. Then what we did risk benefit. The risk is that this may cause cardiac event. The benefit is this is one of the most effective and reasonably priced drug. So should we continue or should we not continue? The decision has to be made on the risk benefit, and what decision was made? The risk is much higher. Even though there is no evidence in India, we should not allow our Indian population to suffer unless there is reason to believe that this patient will, this population will not have this effect, and therefore rofecoxib was banned. The other celecoxib, etirocoxib, nimesulide are still continuing. People must talk of maybe talking about the nimesulide. People say that is banned. No, it is not banned. It is banned in children, because the risk-benefit assessment is much adverse in children. In adult, it is still good to take with caution. So that is how the risk-benefit assessment is always done. You may know anti-cancer drug. Every anti-cancer drug has a serious side effects or long list of side effects, but the benefit is more. Three triangle: risk, benefit, and the alternate drug. And that's why you do the compute the risk benefit assessment and then communicate this to the regulator, to stakeholders, so that they can take what is called as an informed decision in therapeutics. Now collect and record AE, that's one. And what you do is, this is the simple adverse drug reaction reporting form which we have created for pharmacovigilance program. This is also available in website, and this is also available in all ADR monitoring center. It takes some time. What is important in this form? You give the name of the patient, a description, then what has actually happened, the narration, and what is the dose, what is the concomitant medication, and what is the causality assessment in opinion of the person. And then it comes to PVPI, Pharmacovigilance Program of India, and then they collate it, they compute it, and then generate signal, and then that's it. Sources, well, this can be a spontaneous source. Means you have seen from somebody reporting. This can be published literature, PubMed, or maybe Scopus, periodic safety update report. Now recently, I inaugurated in last month a software which was developed by Elsevier, which has the largest periodics journals. They created a software for industry reporting, because for them it is mandatory that Every day they have to scan all journals throughout the globe. What is the adverse effect happened to that drug or their drug? And it is not easy. So this software they have created, they launched in Bangalore, they launched in Hyderabad, and they launched in Bombay. So this has an automatic scanning and then filtering and then cleaning and then give you the data and also doing the causality assessment. So this will help into industry to collect the data of any adverse effect happening to that drug globally and then compute it. So that is the importance of collecting this data. Other methods, active surveillance, as I said, some hospitals, like we have 1,000 centers. We give the 10 centers that now you do this active surveillance for this drug. Nursing, the pharmacist, they are also there. Focused pharmacovigilance is only in India, remember that. We started that because there is an evidence that this drug has caused some side effects, but it caused retinal damage. It's serious, but then it is one or two anecdotal evidence, but we cannot avoid it. Therefore, focused pharmacovigilance, you can do it, you can be participate, can be part of this. Prescription event monitoring, this is, I think, a software has been created in a startup in IIT. They are recording the prescription, collating the prescription, and also assessing what is the prescription event which is happening. Disease registries, we have been talking about the adverse effect or related to drug, what are the pregnancy related adverse event happening. Nowhere the registry was there. So this is the first time under ICMR the registry has been created about the teratogenicity and the drug. We know that this drug can cause teratogenic effect, but there is no registry that what can cause and how many incidences. And then what studies what you can do. This is particularly of interest for the postgraduates. Cross-sectional survey. You do a case control study. You can do the cohort study, and you can do a large safety trial which is done by the primarily with the industry. Drug utilization history. So these are all part of the pharmacovigilance. The causality assessment, as I said, this is done by different methods. What are important thing is the quality check of completeness. Suppose this is an adverse drug reaction which is noted. But if this adverse drug reaction is completed, description is not there, when it happened, when it stopped, what medication were simultaneously given, and then it will be useless. So completeness. And I must say, when we started reporting from India, the completeness score is called from 0 to 1. It was 0.3. But today, the completeness score of pharmacovigilance proof of India is 0.91, which is 0.8 global. That means our pharmacovigilance system has much matured today. Validity of case, perform case follow-up, and then the causality assessment. This is something that's important. Causality assessment and ADR, valid report should have patient information, date of reaction, adverse reaction, suspected medication, and who is reporting. So that there is, so this is, we give training on how to fill up this adverse reaction, and more than 300 training for such pharmacovigilance has been done in Indian Pharmacopoeia Commission. Causality assessment, I will not go into much detail because we will having separate, but more important, Naranjo scale and WHO. These are the two scales which are used. And then code the database, and then you can see that coding events, WHO, because as I said that you have to have a one language so that the computer can compute it easily. Progress, computer-based risk-benefit assessment. I think that is what is more done. Risk-benefit assessment, you can see, if the risk is more than the benefit, then one has to think. And it is important that is one man's meat is another man's poison. So it is risk-based and a benefit assessment, and then linked with the use and to whom. That is what is done in the causality assessment and the risk-benefit assessment. That is the job of experts. And this job of experts, who will do that? The pharmacologists and medicine people together. Signal, as I said, this is a signal we are generating as a signal review panel, and the regulatory action suggested by CDSCO. and the adverse reports of the new drug. Widening scope of pharmacovigilance is not only reporting of the adverse effect, but it is also the medication error is also broad scope of pharmacovigilance. Herbal traditional medicine, sustained substandard medicine, and so this is also under the scope of, these are the drugs which have been banned recently in India, and several Nimosilid under 12 years. So just remember that Nimosilid is not banned completely. Finally, I would say that if you've done so, and the question is, so what? And what next? So it is important that this must be get communicated to those who are using it. And that is what finally I say, communicate the safe if you recommend stakeholders. Who are the stakeholders? The stakeholder is the patient, most important because he suffers. The stakeholder is the policymaker because he has to take decision on the drugs. The stakeholder is the press because the wrong causality assessment publicized that this drug caused a right publication. Public and the physician and medical practitioner and the pharmaceutical industry, this also. So the lag period between detection and regulatory action. So you must remember that if in a clinical trial or if I see that this drug caused serious side effect, I have not done enough homework for that. The regulator has not taken action. People have, large number of people have taken it and there are people die. Whose responsibility is that? I call them is a murder. And who is the murderer? The murderer is all stakeholders who did not record it, who did not report it, who did not take action. So that is something, so it is very important that is lag period between, and that is why when SAE happens, serious adverse event in clinical trial, there is an immediate 24-hour regulatory requirement that you have to give that information. Within 14 days, the causality assessment has to be done. And if it is not done, this regulatory action can be taken. And people say, why this 24 hours? Because so that if there is information to be given to others, then don't take that, it should be prevented. You may be knowing that recently there was incidence of death because of the cough syrup in some countries. So that could have been prevented, perhaps, that if the earliest warning after first death and causality assessment would have been done, you could have prevented the other deaths which has happened because of the same drug was consumed because they were not given the information of the first or the second case. So that is important of that. So this is, I think, this is something which is important, feedback, publication, training and awareness. And that's what I thank the National Academy of Medical Sciences that at least an awareness is created. So that is the reporting practice will improve. And finally, interaction with physician and paramedics. As a pharmacologist, it's your duty, responsibility to go and penetrate with the clinicians and tell them what is to report and then response and the reward. This is hybrid moding. We will not go how to integrate different models. So we have a hotline now. We have ASHA, rural, radio, TV. There's also, you can do that. Educational, PV education. I just made this slide because people attended just like this lectures, seminars, and then they started giving training in the pharmacovigilance. So this was raised in WHO meeting also that in India there are more than 60 pharmacovigilance training bodies. So this is bad. So good is you help sustain reporting culture. Bad is mushrooming of pharmacovigilance courses. And that's why the pharmacovigilance course, which will be started by NAMS, adequate information, reasonable, authentic information, regulatory pharmacovigilance. The ugly is the training by untrained people and the lack of standards. So I put them good, bad, and ugly. So that is the situation which is happening because a lot of, and so where do we stand? Challenges in India, and we skip that. There are so many examples like Nimosilid, hepatic failure, not in children below 12 years, phenylbutazone, metamizole. So these are the drugs banned in India, and the pharmacovigilance in this is, so this is the, these are the centers. Now these are the centers which was earlier, now there are more than 1,000 centers. So this is the pharmacovigilance meeting. So I would say that what is important here is this pharmacovigilance is not the job of a pharmacologist, is not only the job of a regulator, industry, but it is the job of, unless the clinician, unless the prescriber, unless the regulator, the policymaker join hand together, the medicine safety cannot achieve the desired level of satisfaction. And globally, we want to have the reduction in the medication error, a reduction in the adverse reaction, and have a safe therapeutics. So as I said, this is just a sensitization, but we will be having different tests on those topics which I've just mentioned. I would welcome if there are few suggestions or few comments. Maybe our moderator can hand it to her. Thank you very much. And I am extremely thankful to Professor Kapil that he is so patiently listening to a topic which is usually considered as a boring. And of course, to Dr. Sood.
Sir, the issue is, one of the important aspects which you missed is the reputed medical journals. Sometimes they come out with certain publications like statins is useless. Okay, now what data they have, how do they come out of it? But that is being widely utilized by the mass media, and then people remember the incorrect information or half-baked information. So I think that in future lecture, you must take up this issue of wrong publication or what you call very, very, I must say, ki तहलका मचा देने वाले publication. Okay, like
You use correct word.
We had the issue of antimicrobial resistance, and during that time we had that big, what was that, microme we had, which was published in Lancet.
Delhi.
Yeah.
New Delhi strain.
I'm sorry? And then when
Delhi
the
Superbug.
superbug, and
Delhi
when a group of medical microbiologists, they wrote a strong letter to Lancet editorial, then what they did, they made a very small 4-liner regret that data was not based on this much of data. So, but our medical tourism industry was affected.
Yes.
So I think that you must take care of this also in your future lectures, sir. Thank you.
Yeah, thank you very much, sir, for the important feedback. And I would like to especially thank today's speaker, Professor Y.K. Gupta, sir. Sir's presentations and lectures are always engaging. I have been attending sir's lectures since 2017, since I was a student resident at AIMS New Delhi. So thank you, sir. It was an very, very interesting presentation, and this is extremely important for all the researchers and clinicians. So, starting with the questions and doubts, sir. The first one is thank you, sir, for covering the very important topic. Please guide for an authentic portal for knowing the drug interactions in clinical practice, mainly for geriatric population, since there are many apps or websites available, including Medscape.
I would say there is a, if you're asking that what is the no such authentic portal which is official portal for identifying, but you can see what are the adverse drug reaction in PVPI. This is a known adverse drug reaction, but all these portals, they have a lot of information, but you have to basically see that what is the clinically relevant. I always say there may be 100, as Dr. Kapil has also mentioned, there may be 100 adverse drug reaction which are mentioned, which may be coming, but how many of them are clinically relevant? particularly drug interaction, if you just put one drug and then maybe 40 drug interaction. But we have to assess that what are the clinically relevant. And that is what can happen only when our combined effort, maybe in next 5 years, then we can develop Indian data and say that yes, these are the drugs. And particularly in geriatric, where there are multiple drugs, I think that's a very challenging situation. But there is nothing which can be called as an official portal for
Thank you, sir. So next question is from Dr. Sumit. Uh, he asked that we often come across situations where clinicians often ignore reporting the ADRs, maybe because they are afraid to report since its name has vigilance word, and often think that his clinical judgment will be questioned. So, how this mindset can be changed? Please guide us in strengthening the reporting.
This is a very perpetual question and very rightly asked. And we find it difficult, particularly when we give this topic to thesis to our PG or pharmacovigilance associate. He goes to clinician to say that he reported. They are usually reluctant. And this is not India, this is global. In India, it is more. The reason is being that the clinician with the patient has just a 3 minutes average interaction. Now he started reporting, and we say that the form will take 5 minutes, but it doesn't take 5 minutes, it takes more than that. Now that is one. Then he thinks, what I am getting out of it? So we don't have any answer to that, except that you will be contributing to the academics. That's it. And third thing is the belief that this is a pharmacovigilance. If I report 20 cases, then I may be found guilty, or people may come and complain that this person is creating, this therapy is not good. So we have to tell them the reporting of adverse reaction is out of legal purview. Medical negligence and ADR, they are two different things. This is what the education awareness workshops we teach. That reporting, if you reported adverse, this is just like happening what you see in an accident or murder, not accident, a serious accident in roadside. People now have started reporting it. But earlier, the reporting will not go to the police station because they will found that they will get involved, they will call. So we have to similarly we give here that pharmacovigilance reporting adverse reaction has nothing to do with the patient or nothing to do with the physician. The negligence is different, but so that has to be very clearly told to them, and that's what the workshops are required.
Sir rightly said that one need not to fear and the negligence will be counted upon. So the next question is from Dr. Sri Sudha. So Dr. Sumit and Dr. Sri Sudha both are faculty of pharmacology, and so they are actively working on pharmacovigilance. I have seen their work. So Dr. Sri Sudha asked, good afternoon, sir. Can you tell what are some recent medical devices recalls due to material vigilance findings?
Important points, Shri Sudha. Except the Johnson hip joint, which was recalled, there was a batch of the intraocular lens which caused severe reactions. That again was the credit goes to 1 or 2 centers in pharmacovigilance which are doing, and this was reported, then this was collected for pharmacovigilance, and then that batch, that entire was recalled. And then we are looking for the pharmacovigilance of the catheters, particularly the catheters are given which are the cardiac catheter, coronary stents, and some data is being generated, and some have been not withdrawn, but they have voluntarily recalled. But when we voluntarily recall, they give half of the information. But the practical thing is the incidence of rethrombosis was more. Getting such data from devices is much more complex than the drug, because it has a lot of connotation, confounding factors. In coronary stent, what we see, restenosis is one cause. And restenosis, if it is happening, it happens in, can happen in any case with any device. But if the restenosis happens in larger number than expected, then you can assess the cause. So this is what, and also I will give you the complexity of the coronary of the Johnson and Johnson example, because that is a case study of material vigilance for the hip joint. This, if the joint failure, replacement failure can happen because of, when you say replacement failure, means re-surgery requirement. Re-surgery requirement can happen because of the individual case, because of the surgeon incompetence or less experienced surgeon, and 1, 2, 3, 4, and then fifth is also the poor quality of the material. So now this has to be very carefully assessed, and for which the completeness of the data information is important. So these are some important things. But I would say not many, except these few 3 examples, have been recalled because of the material vigilance. And that's why important thing is people should work on material vigilance.
Thank you, sir. So the next question from ma'am is, the primary challenge we face universally is the lack of reporting. So would it be possible to implement or introduce specific postings for MBBS interns to encourage their involvement in material vigilance or pharmacovigilance, since they are posted in all the departments and they are going to be a future doctors, clinicians, researchers?
But then if you say that somebody is only posted for pharmacovigilance, material vigilance, I think that would be will not be possible because they have a very limited time. But it is important that when they are posted in clinical disciplines, they should be also told as one of the objectives to collect adverse reaction, because they spend more than 6 months in clinical disciplines, and that is the time when one of the logbook should have that how many cases you have reported. And this we tried at few places. These type of innovative, very effective ideas only work when there is a champion, whether it is director or head of department. When they think yes, it should be done, then it will be done. When they think yaar isse kya hota hai, ye to hamari galti nikalne ke liye aa gaye, so it doesn't work. So this is but very valid point.
Thank you, sir. Sir, another question. If an implant fails without any fault on the doctor's part, how will the medical legal issue will be addressed?
See, the medical legal issue and is because if there is a fault of, this is exactly the medical legal, I am talking of the hip joint. This became medical legal because person complained it and there was no fault of the doctor. Then it has to be a committee-based decision that who and how it is decided that is. And then it was decided that it should be given compensation. The company fought in the court that why I should be given, but then court said no, if this is to be given, then given. And then it was a global phenomenon because in multinational companies, so they had agreed. So such thing ultimately result into litigation, because when there is a compensation of and goes into 10 crores, 20 crores, 100 crores, then definitely they will fight tooth and nail that this is not related. And that's why causality assessment skill has to be very strong. This is just like a murderer will got scot-free because of lack of evidence. And the same thing, if there is a causality assessment is not done correctly, then such things are happen. This is very.
Thank you, sir. So again, causality assessment is very important, and thanks to the speaker, thanks to the participants for asking interesting question. Sir, one question from my side. Sir, I had a query. Since you told that from patient to policymakers, all are the stakeholders in pharmacovigilance, and the press is one of them. So, sir, are there any regulatory guidelines for medical misinformation through media and social media? Since nowadays,
Unfortunately, not so. And that's what Dr. Kapil said, that if there is a media hype of some adverse reaction, then, or see, I will just give you one simple example. Maybe when about 6-7 years back, in Bombay or in Maharashtra, there were several cases of hepatotoxicity because of paracetamol. Several. Now, this was in newspaper. Now, suddenly people got worried about what happened to paracetamol, why suddenly there is. And then, after 15 days, there were several cases of bleeding, peptic ulcer, and all those because of the anti-inflammatory, antipyretic drugs from the other part of the country. So this was a corporate war. Ultimately, this was exaggeration or of selective, and this was found that, of course, there is no evidence. This was what we found, that this was because of the promoting paracetamol. This group was promoting this because paracetamol was considered to be the best and safest drug. So people said, "Yaar, paracetamol khaoge to ye ho jayega." So these things happen in corporate war. So you have to be very careful about the analysis of what you call is a half-baked, half-cooked, or say ardha satya information from media which you get.
Thank you, sir. You correctly mentioned that these are the satte. It affected the patients, like in the war of corporates. It ultimately caused the suffering of the common people and the patients. So, sir, that was an amazing session, I would say. So now I would like to request Professor Ajay Kumar Sood, sir. Sir is Deputy Secretary of National Academy of Medical Sciences. So I would like to request sir for the
I will also request before Dr. Sood says that I have given you the list of pharmacovigilance topics which we will be covering. You may give the feedback to Dr. Kapil and Dr. Sood so that we can plan accordingly. With your feedback will be very useful.
Sir.
Thank you so much. On behalf of the National Academy of Medical Sciences, it is indeed my great privilege and pleasure to thank the distinguished speaker for this afternoon, Professor Y.K. Gupta, sir. Sir has been out of Delhi and he was in Bangalore, and he came for this session directly from the airport. And we are really grateful to you, sir, for your discussion. And in this particular session, sir has again made a commitment that he is going to be with us for 5 more sessions. So that is a great thing, and we really appreciate your efforts, sir. And this session has been very, very important for all of us, as he said, right from the patient to the young doctors and finally to the policymakers. And all of us have to make a little bit contribution to create awareness. And I would like to place on record Dr. Gupta sir has carried more than 300 workshops all over the country for sensitization in the last few years. So he's making a lot of efforts, and I think this topic is very important for clinicians, not only for their clinical practice, but also it will help you to become better sort of physician. And he mentioned that many of the institutions have reporting units, nearly 1,000 units are there. So if there is a reporting unit in your hospital or in a medical college, so be in touch with that unit, and that unit can further guide you and give you more orientation about this program. So thank you so much, sir, for giving us very informative talk, and all of us have really enjoyed and learned from that. So I also thank all the faculty members and postgraduate students who have attended this program and made this program very, very interesting and interactive by asking questions. So it is your own program. And as sir has said that he has given us a list of topic on pharmacovigilance. If you want to add or modify that list, or even if you can come forward to volunteer to take some of this session, you're always welcome. So your suggestions for the improvement of this program is always welcome. You can always write to our secretary, Dr. Kapil sir. I will also like to inform you that our next session is due on 7th of March. And there again, we have a very eminent speaker from All India Institute of Medical Sciences, New Delhi. And in this session, we are going to cover autism spectrum disorders. So this could be again of interest, especially to the pediatrician and to the postgraduate. So please do attend that program. We'll circulate you all the details. In case you have some specific question for the speaker, if you share this question, we'll communicate this to the speaker. So one more information that as NAMS in collaboration with IIIT Hyderabad has started this online course on artificial intelligence in health. And Dr. Y.K. Gupta is one of the committee members who has recommended that this course will be continued in 2025 also. And all of you who are interested, especially the young faculty members and the postgraduate student, you can always refer to the NAMS website or IIIT website for registration. The second batch is likely to be started in the month of March or early April. So that is a very important, I think, technology which is going to revolutionize the health sector as well. Another information is for postgraduate student. In collaboration with ICMR, we have a biostatistical helpline. So this again is an online facility for the postgraduate student, whereby your question related to sample size, sampling technique will be answered by the eminent statistician, both from NAMS as well as from the ICMR. So please look into the website, NAMS website, as well as the ICMR website. You can always book a slot on that, and you will have an opportunity to interact with the best biostatistician for 20-25 minutes, and it can be repeated. So please make use of these facilities as well. Once again, Dr. Jaswant, welcome back to Delhi. Earlier he was at AIMS Dioga, but recently he has joined Delhi. So he has been helping us, as you know, from the very beginning as a moderator and doing this job very well. We are extremely grateful to you for your support to the Academy, and we look forward this will continue. Here in our office, Mr. Ravi, who is our IT officer, he is there. And Mr. Shivam, they have been helping us writing you letters, reminding you again and again on emails and sending you the Zoom link, and also ensuring that this is telecast on the YouTube. And these sessions will be put on our website in the form of virtual library. So thanks to Mr. Ravi, Mr. Shivam, and the entire team who has worked in the background at time. So once again, I thank you all for being with us this evening and making this evening very interesting for all of us. Namaskar. Thank you very much. Over to you, Dr. Jasmine.
Thank you, sir. So I think with this we can mark